drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. If you like to read like I do, then you should check out the Rewires app. It's totally free to download, and if you go through the link in the episode description or on my support page, you can get a free 60-day trial for zero cost. Rewires is a really cool app that allows you to import digital highlights from over 30 different platforms such as Kindle, iBooks, PDFs, and more. It makes it really easy for you to review and find and share excerpts and quotes from whatever it is that you're reading, whether it's PDFs of scientific papers or books that you're reading on your Kindle, and you can even save and use highlights from physical books. I read a lot of physical books before bed, for example, or when I'm traveling, and the Rewires app actually integrates with the camera on your phone so that you can take a photo of a page that you're reading, highlight a specific chunk of text from that page, and export it to the app so that you can reference it or find it or share it later. They even allow you to export really cool looking graphics of some of the highlights that you have in the books or the papers you're reading so that you can share them in an engaging way. So check out the Rewires app today if you are a reader and you like to reference and organize what you read. Go through the link in the episode description or on my support page or in my free weekly newsletter at mindandmatter.substack.com and try the Rewires app 60 days free today.
Thanks for having me.
Do you want to start off by just telling everyone a little bit about who you are and what your lab studies?
Hi, I'm Jonny Kohl. I'm a group leader at the Francis Crick Institute in London, UK. I started my lab there about 6 years ago. And the official title of my lab is the State-Dependent Neural Processing Lab. And that really describes what we're doing. We're trying to figure out how the state of the body affects brain form and function, and using mostly mice as a model system. Yeah.
And you study a lot of instinctive behaviors in mice.
That's right, exactly. So we are really into instinctive behaviors. So those are behaviors that animals can perform with very little or no prior learning. And why do we care about those behaviors? I find them personally really fascinating because they're the sort of the behaviors that animals come with. So they're pre-wired into their brains. And basically, allow them to, perform very complex actions that they, in order to cope with the challenges that they normally encounter in their ecological niche. For instance, instinctive behaviors such as escaping from a snake is something that mice can do instinctively without having to think about it too much, because these are behaviors that you don't have the luxury to learn. Because if you can't really you can't afford to make any mistakes there, and that's why they're pre-wired into the brain by natural selection. And why are they so interesting? It's because they have a strong genetic component. They are developmentally wired into the brain, so we can really study, we think, the relationship between genes, between circuits, and between behavior, between all those three levels, or very well using those behaviors. Yeah.
Mm-hmm. And so I want to talk about instinctive behaviors a little bit at a high level. On the one
Yeah.
hand, we have these behaviors that are instincts, so they're baked into the brain. The animal doesn't really need to learn them, but that doesn't necessarily mean they are simplistic or purely reflexive, like tapping on your knee. They can be really complex and elaborate and modifiable by learning and experience, even though they're pre-configured.
Exactly. That's actually one of the most fascinating properties of instincts that on one hand, they're supremely robust, because as I said, you can't really afford not to escape from a predator the first time you encounter them. At the same time, you can't afford to not learn. If you're repeatedly exposed to the same, sort of stimulus, for instance, and you have to learn at some point that this might not be dangerous. Otherwise, you're living in this constant state of alert, which is not beneficial. So at the one hand, you have these very robust behavioral outputs. At the same time, these circuits, like any other circuit in the brain, can learn, can be modified by experience, but also by internal states. And this sort of dichotomy is really interesting, I think. Yeah.
Mm-hmm. And can you just give us a sense, give us kind of a survey for some of the major instinctive behaviors that we tend to see in mice and other mammals? So, for example, there's instincts related to eating food and finding nutrition. There's instincts related to parental care behavior and so forth. What are some of the major sort of clusters or families of instinctive behaviors that we tend to see in most mammals?
So I guess as you said, there are instinctive behaviors related to finding food. That starts out when you're a neonate. You have to find your mom's nipple. You have to start feeding pretty much instantly because otherwise you'll be dead within the space of a few hours. Then you have to seek warmth. You have to learn to not learn, but you have to take action to regulate your temperature. You have to avoid and evade predators. You have to later in life learn how to find a mate, and you have then even later learn and in a way to look after your young. And these are behaviors that have very strong instinctive components. So if you look at mice, for instance, they can take care of their young pretty much the first time they see them. They obviously don't have any antenatal classes. They just have to perform right out of the box. And that's extremely fascinating because these are not simple behaviors. They're extremely complex behaviors that these animals can perform pretty much the first time.
Yeah, and a lot of these instinctive behaviors depend on context or depend on the state of the animal. So the animal's not just initiating these instinctive behaviors all the time haphazardly. They're happening at the appropriate time in response to certain stimuli. Sometimes they don't happen even in the presence of a stimulus that would otherwise cause it in a different context. I want to talk a little bit about this concept of state. So your lab studies state-dependent regulation of behaviors. What exactly is state or behavioral state in an animal, and what are some of the major inputs that define what that is?
Yeah, I think that's a very good question, and it's a very, in a way, a very, contentious point still because different neuroscientists have very different definitions of state. There are people that say that states are states of the brain that change on a millisecond time scale. What I mean by state and what sort of my subfield means a state are sort of internal states that unfold over longer time scales. We're talking minutes to hours to even days and weeks when you talk about hormonal states, for instance. And the states that, for instance, my lab is extremely interested in are these hormonal states, so these slowly changing states. For instance, if you look at pregnancy, that's a hormonal state that unfolds over the course of many days and weeks and has profound effects on brain processing. So that's what we see as states in the lab. Yeah.
Mm-hmm. And so when we think about states that unfold over minutes to hours, we can start to think about, just everyday things in our own life. Everybody gets tired eventually and they get sleepy or they become more aroused or more energetic. Everybody gets hungry and thirsty and so forth. And we all know from experience, these things don't happen instantaneously for the most part. They take minutes or hours to unfold. There's a persistence to them. They last for some amount of time. They don't just go right away for the most part. They take time, and there's sort of a time course and an evolution to these things that's on the minutes to hours time scale for a lot of the ones that are the most familiar. Your lab, one of the things you study is hunger. Let's talk a little bit about that because you study it, but also because everybody's familiar with it. Presumably, when I was a little baby, I didn't have to learn to be hungry. I just had this uncomfortable feeling inside of me. And maybe over time, I learned what to do when I get hungry or what will make me not hungry anymore the fastest. But this feeling is sort of pre-configured and it's inside of us. So when you're studying hunger in the lab, can you talk a little bit about what we know about that at a high level in terms of where this state or this feeling or whatever you want to call it comes from in the animal, in terms of the brain regions involved and some of the hormones and peptide systems that regulate the time course of this stuff?
So I have to just one disclaimer. So we are not like bona fide hunger experts. We just recently made a foray into how hunger affects our favorite behavior, which is parental behavior. But during this project, we had to sort of learn more about the neural basis of hunger. And it turns out in recent years, the picture of how hunger is regulated in the brain has become increasingly complex. But there are a few principles. For instance, there are specific groups of genetically specified neurons in the hypothalamus, so a highly evolutionary conserved area of the brain that regulates homeostasis, internal states. We can talk about what that actually means. And there's a few small groups of neurons. For instance, these neurons called AGRP neurons in the arcuate nucleus. And they are, for instance, they're called the hunger neurons because they detect caloric deficiency. And then what they seem to be doing is that they sort of encode or set up an averse motivational state, so what you can, what is the feeling, this negative feeling of hunger. And they then regulate all of the actions that are necessary to get rid of that feeling, and that's seeking food, that is, I guess, suppressing other types of behavior. And for instance, when people record from these neurons, what they see is that as the animal is food deprived, the activity of these neurons ramps up and up and up and up. And then when the animal even sees food or smells food, the activity of the neurons is very rapidly then suppressed. So it's not the act of actually replenishing your energy stores or feeding. It's already the anticipation of that happening that suppresses these neurons. So that's in a way a very powerful combination of an instinctive behavior that is informed by the learning of what happens if I see food. I'm going to consume this food. So this is in a way an anticipatory regulation, and then that's a theme in hunger circuits. There's not really a homeostatic system that only reacts after the fact. I'm calorically depleted now, so now I'm taking action. There's actually what we call an allosteric system that sort of anticipates challenges, because once your energy stores are low, it's basically too late. It would be a very dangerous situation, So it's a system that's constantly trying to anticipate what will I need if I continue exerting this effort, for instance,
Yeah.
and that then drives these food-seeking behaviors.
So there's neurons deep in the brain, literally deep in the middle of the brain, in a pretty conserved part of the brain called the hypothalamus. And these neurons are more or less dedicated to what we would call the hunger state. And there's a lot of nuance there, but hunger is this sort of thing that's baked into the brain. These neurons are an important part of it. These are highly conserved neurons in many ways, but importantly, they're responding and doing things at multiple time scales, not just the slow replenishment of calories. They're responding at fast time scales too. And I guess we might connect that to our everyday life. You can be really hungry, and then as soon as you see your favorite restaurants or you smell the food, your hunger, you can kind of forget about your hunger, even though you haven't actually swallowed the food yet.
Exactly. So these neurons, all of these neurons in that part of the brain, they're really multiplexing. So that, for instance, they have different types of neurotransmitters, or we could call them neuromediators. They have fast neurotransmitters such as GABA, so that they signal over the course of milliseconds to downstream neurons. But they also release neuropeptides. Two different types in the case of the AGRP neurons. So the AGRP peptide itself, but also the NPY peptide. And those peptides, they really signal to downstream circuits on the order of hours, minutes to hours, and even half days and days. So thereby the same neuron can basically participate in these different functions that you just mentioned. Yeah.
Mm-hmm. And another thing that's interesting to think about, and that your lab has studied, is, okay, we've got this circuitry in the hypothalamus. It can respond on multiple time scales. In some ways, it's dedicated, we might say, to hunger, but at the same time, it has to be modifiable by experience, and it has to really hook into other motivations in important ways. Again, we can think about just we can think about this intuitively in terms of our day-to-day life. A lot of us, when we get very hungry, we become very irritable. We become more aggressive, basically. We're more likely to lash out at people. We're more likely to take risks, to go out into the environment, to actually find food to satiate this hunger that we start to feel. And so you've done some interesting work recently that sort of ties into how some of these different motivational drives interact with one another. Because on the one hand, because we all have these drives, we get thirsty for water, we get hungry for food, we might get lonely and want to find a mate, we might get tired and need to go to sleep. But all of these things, can be happening simultaneously and in parallel. And somehow the brain has to integrate all of these things and kind of decide in any given moment, which motivation do I prioritize? Do I go get food right now and risk danger, or do I go look for a mate, or should I take a nap first? So how did you start to get thinking in this direction about how these different things integrate together at the level of, say, the hypothalamus?
Exactly. I mean, that's exactly one of the key questions that we started asking a few years ago. So if you think about internal states, there's dozens of them, maybe hundreds. You have this multitude of states. There's this sort of state space. But then at any given point, an animal only has a few behavioral choices to make. For instance, if you're confronted with an infant, with a pup, in the case of a mouse, you can take care of it, you can attack it, or you can ignore it. So you have this, very high dimensional state space, but the behavioral decision space is very low dimensional. So what we're wondering is how this high dimensional state space maps onto these only very few behavioral decisions. So where in the brain this integration happens, so where is the decision point made that, okay, I'm hungry, it's early in the morning, I'm in this part of my estrous cycle, and so on, I'm stressed, and so on. How does this all get integrated, and how does it inform behavioral decisions? And we used a very simple model system to study how different internal states are integrated. We used just virgin female mice, so sexually inexperienced mice, and expose them to pups. And normally what you have is you have a certain percentage of these animals that basically ignore these pups, and a certain percentage that is spontaneously parental, at least in lab populations.
You mean spontaneously?
Spontaneously, because I guess they're not parents. They have no reason to be parental, but they spontaneously exhibit behavior that looks very much like parental behavior. So they retreat to the nest.
You're talking about like a female animal who's not been pregnant herself. She's got no experience with pups. But if you put them in front of pups, some percentage of the time they kind of act like mother mice, and some percentage of the time they're more aggressive and they do other things. And it just depends. But there's no prior learning involved. And there's this kind of chance or probabilistic component to what behaviors get expressed.
Exactly. That's what we usually observe, And then one question was, what actually, sets that probability? And then, we made the serendipitous discovery, and that's really what it was, that when you food deprive these animals for only a short amount of time, we're talking 3 hours to 6 hours. That's not even in an animal license. That's not even classified as a food deprivation. What you start seeing is that more and more of these animals become aggressive towards these pups, and that's not something that you normally see in the sated state. And that really got us, I'm thinking, okay, so how is this interaction between the hunger system and the, parenting or pup-directed behavior system happening in the brain? So there was one layer of this project, and we dug into the circuitry and found that there's this, quite direct interaction between these hunger neurons that I mentioned, these AGRP neurons, and a key node in the parenting circuitry. So these neurons in the so-called medial preoptic area. And then after that, we found that there's another layer of regulation here, namely the state of the estrous cycle that each animal is in, then also sets the probability with which they show aggression after food deprivation. So you have these two very different states. We call them orthogonal because they are seemingly independent of each other. So you have estrous state, hormonal state, but also hunger state. And we found that those two states have this very intricate interaction in neurons in the hypothalamus, so in this preoptic area.
I see. So you have animals that have never been a parent before. So these are female mice, but they've never been pregnant. They never had their own pups. You put them in front of pups, and basically what you're saying is if all of their other needs are met, if their stomach's full and they're not hungry, they're more likely to be nice and act like mommy mice to the little baby pups, even though they've
Right.
never actually been a mother themselves. But if they're hungry, they're much more likely not to do that and to either ignore them or even be aggressive towards them. And so we know, you already knew, that there's basically separate circuitry in different parts of the hypothalamus. One of it's more focused on hunger and food stuff, and one of it's more focused on social behavior and parental behavior stuff. And they're separate at one level, but there must be some kind of interaction here, because the hunger state of a female can dictate whether or not she engages in maternal or parental care behavior or not.
That's correct. Exactly. That's exactly what happens. Yeah.
I want to give people a sense, before we kind of dig into those particular results, for how some of these hypothalamic circuits are regulated by hormonal state. So if we just talk about the estrous cycle in mice, can you kind of describe that at a high level for people? Try and connect it to, the monthly cycles that human females experience. And importantly, how are the hormone fluctuations actually getting in the brain and affecting behavior via circuitry?
Absolutely. So in contrast to humans and other mammals, which have estrous cycles, so there's a shedding of tissue, there's menstrual bleeding and so on. Mice only have an estrous cycle, so that comes with certain cellular changes in their reproductive system, but it's not as dramatic. And the estrous cycle it has a duration of about 4 to 5 days and has 4 distinct phases. And it really comes with fluctuations in several different types of hormones. But the major players are, what we know from humans as well, are estradiol and progesterone. So those are the two major players here. And they show these very characteristic fluctuations across the estrous cycle. And importantly, and it's important for our study, for instance, the relative ratio of these hormones also changes throughout the cycle. Now, these are steroid hormones. So they're derived from cholesterol, which means they are sort of fatty molecules. And as fatty molecules, as lipophilic molecules, they can just diffuse wherever they want. They originate mostly from the periphery of the body, from I guess the reproductive organs, and they via the bloodstream can just reach every little corner of the brain. So there's no really a notable barrier to the diffusion. And that's if you're a neuron in the brain that expresses a receptor for either of those hormones, we can be quite certain that you will react to those hormones at some point during the estrous cycle.
I see. So estradiol, basically estrogen, the main estrogen in the body, progesterone, these are two key hormones that are fluctuating across the estrous cycle. They're fat soluble, so they can diffuse into the brain. They can get into neurons in different parts of the brain. And as the relative ratio changes across the estrous cycle, they're getting into the brain in different amounts. That ratio is changing. And then once they're in the brain, how are these things actually affecting behavior? How do they change neurons?
Mm-hmm. They do that in the most interesting ways, really, because they affect neurons at totally different timescales. So what they can do is they can directly affect neuronal physiology at very short timescales by binding to receptors that are situated in the membrane, so dedicated receptors. But also they can bind to other types of neurotransmitter receptors, the GABA A receptors, for instance. So they can change neuronal physiology at very fast timescales. We're talking, again, milliseconds to seconds. But then in addition, and that's the more relevant part here, they bind to specific receptors that are called nuclear receptors or cytoplasmic receptors. And these are molecules that are basically shuttling between the cellular nucleus and the cellular cytoplasm all the time. But once they bind one of those hormone molecules, they are recruited to the nucleus and then act as transcription factors, which means that they can directly change gene expression across the genome. And people have shown that, for instance, the estrogen receptor that binds estradiol can change the expression of thousands of genes in a very long-lasting manner. And that's really interesting, because that is sort of a biological substrate by which these two hormones can change neuronal physiology over days and weeks and sometimes even permanently.
Yeah. So we can imagine, so these things change not only month to month, day to day in the context of something like the estrous cycle, but they're changing developmentally, right? When puberty happens, when menopause happens, they're changing when a female becomes pregnant. And so they can affect neurons immediately. They can sort of change gene expression to affect neurons over the course of hours and days. They can also kind of completely resculpt circuits and change things potentially even permanently, after a woman gets pregnant or after you go through puberty or what have you. So multiple time scales with which these things affect neurons, including physically like rewiring them.
Exactly, exactly, and that's why they are so interesting for us as well, because they really they have the potential to basically create these plasticity windows even in the adult brain, and this is what we see in our prior work and during pregnancy, for instance. During pregnancy, you have these crazy increases in the levels of progesterone and estradiol, and what we think is happening is that during these massive increases, they again open up a window of plasticity during which parenting circuits are remodeled, for instance, in anticipation of the animal needing to perform really robust parental behavior. So that's the adult brain, which is commonly thought to be, rather, static and less plastic as the developing brain. But the changes that we see there are really comparable to what's happening in the developing brain.
And so if we step back for a moment, we can think about things like sex hormones that fluctuate across the estrous cycle and development, right? They go up, they go down. We can also think about hunger and satiety hormones coming from the gut. When you're more hungry or more satiated, these hormones start to change. So there's this whole soup of proteins and molecules and things, all of these different hormones, they're going up, they're going down, day and night, week by week. And, the sort of the integrated average of all these things, the sort of profile of that goo at any point is going to dictate what neurons are likely to do or not do. And so this is how we can maybe start to think about what sets the state of the animal, how likely it is to engage in certain behaviors, is sort of like the sum total of all of these hormones and other factors that regulate how excitable or that regulate what the neurons are going to do or not.
Exactly. And that's even, that's already super complex and high dimensional. But then those are even just the sort of the wireless connections, but then you have the wired connectivity between the brain and the rest of the body as well. The vagus nerve, which, I guess informs the brain of the state of the body at any given point in time. So you have this really, dense interaction between the brain and the rest of the body, using wireless and wired connections.
Mm-hmm. Yeah, I think that's a good analogy. So wired versus wireless connections. So neurons are literally hooked up to each other like wires. But what you're saying is basically a wireless connection would be if you have a hormone that like estradiole's high, maybe progesterone's low, so the ratio is really high in one direction. Those hormones are diffusing into the brain, and they can separately in two different parts of the brain, far away from each other, affect different circuits in parallel. So we can think of that as a wireless connection.
Exactly. I think that's how we're thinking about this. That's why these, sort of broadcast messages are so powerful, because they can, as you say, they can, sort of synchronize or coordinate very different circuits that are located in different parts of the brain, which are not even directly connected. So it's a very powerful layer of regulation there that we are not really understanding yet. But that's sort of my lab's goal, is to also understand the principles by which hormones coordinate circuit-wide activity to then, provide different behavioral output.
Yeah, and so at a high level, it starts to become intuitive to think about what you mentioned earlier. So you can take a naive female mouse who's never been a parent, she's never been pregnant, you can put her in front of pups, and if she's hungry, she's not likely to take care of them like a mother mouse would. And you can start to think, okay, well, maybe her parental control network hasn't been turned on because she's never been pregnant, but she's also hungry. So her gut hormones are at one level, and she's in one phase of her estrous cycle, and maybe the estradiole's low or high, and that's going to affect the parental circuit in a certain way. And so this soup of hormones and other things makes her go find food and eat it circuitry more active, and her go take care of the pups circuitry less active. Can you start to talk about these results a little bit and how you kind of got into this study?
So I guess, as I mentioned, it started out with, a serendipitous discovery, namely that I guess just, food depriving animals for a relatively short amount of time basically just makes them, attack pups, and that was very surprising. And then we mapped the sort of the interactions between the hunger circuits, specifically those AGRP neurons, and a key node in the parenting circuits, the so-called medial preoptic area, or MPOA. We found that there's a direct connection, a peptidergic connection between the AGRP neurons and these in these MPOA, parenting promoting, sort of promoting neurons. That is inhibitory. So it's an inhibitory peptidergic connection. So when the animal is hungry, these neurons become more active and they release an inhibitory peptide that somehow seems to suppress the activity of these parenting promoting neurons. So that was sort of the first layer. So that's basically just a very direct connection. And then, as we discussed, we also found that the probability with which these animals become aggressive after being food deprived is completely dictated by the estrous cycle. And so that really suggested to us, okay, so maybe it's either estradiol or progesterone that somehow interacts with either of those nodes in the circuit. So either these AGRP neurons or their postsynaptic MPOA neurons.
Yeah. So, to make sure I'm following, it sounds like you're saying when the female mice are hungry compared to when they're sated, they're more likely to attack the pups. But the specific probability of attack or aggression depends on the estrous cycle. So I'm just going to make up the numbers. These aren't the numbers in the paper, but maybe at one phase of the estrous cycle, they attack the pups 80% of the time. But another phase, they attack them 60% of the time, even though they're equally hungry in both cases.
Yeah, it's actually quite close. The upper limit, as you said, is around 80%, but the lower limit is even more drastic. It's 30%. So you have this more than twofold difference between estrous cycle stage A versus B. So for instance, during estrous, which is one part of the estrous cycle, they are extremely unlikely to attack them even when they're food deprived for more than 6 hours. But during mid-estrous, a different the phase of the cycle, they are 80% likely to attack those pups even after 3 hours. So then the question became, okay, what is it? Is it estradiol? Is it progesterone? So when we basically tried to run a correlation between the levels of the blood plasma levels of estradiol or progesterone against the probability with which they attack, we didn't find any good correlation at all. But then Mingran, the student who was spearheading the study, a PhD student, she had the clever idea of just looking at the relative ratio of both hormones. And then looking at the progesterone to estradiol ratio, that really gave a nice correlation. And that suggested that there is some integration happening of those two hormones in either of those locations in the circuitry. And we then tried to find out where in the circuitry this integration happens. And we did this by genetically removing the receptors for either estrogen and progesterone and see what happens. And that suggested that this hormonal integration happens in these MPOA neurons, so the ones that are promoting pup-directed behavior. Yeah.
And so one thing I want to be clear about with people is, so these hunger neurons in the hypothalamus, these AGRP neurons, they project to multiple parts of the brain and they're inhibitory. So when they release their transmitters and their peptides and stuff, it tends to inhibit the next neuron in the wired connection rather than excite it. So to compress some of that literature for people, one way you can start to think about some of these circuits, including this hunger circuit, it's almost like instead of promoting feeding per se, instead of actively doing that, they're almost promoting feeding by inhibiting other behaviors. Is that one way to think about it?
That's one way to think about it. So that's how it might work in some of their downstream regions. But also what tends to happen in a lot of brain areas, that you basically, by inhibiting neurons, you can release a behavior by so-called disinhibition. For instance, this inhibition is formed onto another inhibitory neuron in a downstream area. You can sort of release the brake of what's happening downstream of that inhibitory neuron. So you basically effectively excite a downstream neuron via inhibition. And the advantage of that is that you have more regulatory control over something that is over something compared to what's called a feed-forward excitatory connection, where you just basically drive. Yeah?
Right. So it's as if in certain parts of the brain, the pattern of neural activity that would give you the behavior, that would release the behavior, it's there, but it's being sort of inhibited at a consistent basis. And you can only sort of release that inhibition if you then inhibit the inhibition. So it's like, -1, multiplying two negative numbers to get a positive number. This behavior is there. This pattern can be released, but it's not going to unless something else comes in and turns off or shuts off the brakes, as you said.
Absolutely. That's, for instance, how the midbrain seems to work, which is, I guess, downstream of a lot of these hypothalamic neurons in the control of instinctive behavior. So an area in the midbrain called the periaqueductal gray, a massive area that is still very poorly understood. And all of the incoming connections from the hypothalamus, which I guess encodes these motivational states onto these midbrain neurons, are inhibitory, or mostly are. And what seems to be happening is that there's instinctive motor actions released through the midbrain exclusively via disinhibition. So that's a very common principle there. Now with these AGRP neurons, they seem to work via disinhibition in some areas, but also it's important to note that these peptides that they release, so NPY and HRP, for instance, they can be inhibitory in some contexts, but they can also be excitatory in other contexts, and really depends on the type of receptor that they bind and the sort of intracellular consequences of that binding, which can differ per neuron. Yeah.
Mm-hmm. So, what was sort of the punchline in terms of how you figured out that estradiol and progesterone and the ratio was tying into this hunger and parental care circuitry? How did the ratio of these things actually hook into this?
Yeah. So what we found was that when we looked at the sort of in each experiment you have you sort of have a number of animals, and each of those animals is just statistically in a different estrous cycle stage.
Right.
Know all of those. We know all of those stages, and then we can basically compute a total probability of the entire cohort of animals with which the cohort becomes angry towards pups after food deprivation. So we look at that sort of expected probability, and we compare it to the observed probability after we manipulate these hormone receptors in either of those two brain areas. Now, for instance, what we observed is, so again, I just need to backpedal a bit and basically just emphasize that what seems to be important here is the ratio of progesterone to estradiol.
Mm-hmm.
Now, when we remove the estrogen receptor from these neurons, that basically that makes these neurons blind to estrogen, and that basically, from the neurons' perspective, dramatically increases the progesterone to estrogen ratio, because now they only sense progesterone but not estrogen. Mm-hmm. And then what we see is that we see a dramatic increase in the observed probability with which these animals become aggressive. So it really scales with this with this expected relationship between the ratio and the aggression probability. And when we did the opposite, so when we removed the progesterone receptor, now the progesterone to estradiol ratio, as experienced by these neurons, is actually much lower. And hence, the probability with which they show aggression is also much lower. And that really told us that in fact, it is the ratio rather than the individual levels of these hormones that are being sensed by these neurons.
Right. And remind me, which neurons are we talking about? Are these the medial preoptic area neurons?
Exactly.
Got it.
Yeah.
So the hunger neurons are in the arcuate nucleus. They're projecting to this part of the hypothalamus. This part of the hypothalamus has the estrogen and progesterone receptors. And so these neurons in this part of the hypothalamus are integrating a sex hormone signal that's telling you where you are in the estrous cycle, together with a hunger signal that's telling you how hungry you are.
Exactly. That's how it seems to work. Exactly. Yeah.
Interesting.
And then the question is basically, okay, how do those completely different signals, how are they actually integrated? What happens inside those cells? And in order to find out what's happening there, how these neurons are capable of integrating those two very different signals, we turn to a technique called patch clamp electrophysiology. So that's basically a technique where we take acute brain slices from a mouse brain, and then culture it in a nutrient-rich medium for a few hours, and then perform electrical recordings from individual neurons. And these recordings are extremely sensitive. They can really detect very small biophysical changes in these neurons, electrical changes, but they can also allow us to then afterwards reconstruct the morphology of these neurons. So it's a very labor-intensive but powerful and very sensitive technique. And what we found was that both of these different signals, so the hunger signal and the hormonal signal, they converge on a single ion channel. So basically a pore in the neuron membrane that regulates ion flow into the cell and regulates the cell's excitability. And both of these factors, these signals seem to be converging onto one such type of ion channel, the so-called HCN channels.
I see. So putting this stuff together, basically what it sounds like is throughout the estrous cycle, hormone levels are fluctuating, the relative ratios are changing. In particular, a key ratio here is the estradiol or estrogen to progesterone ratio. As these two things change, right, one goes up, one goes down, the ratio is changing, these hormones are getting into the brain and the ratio is changing, and both of them can get into these neurons, and the relative ratio will dictate how much of this ion channel gets made in these particular neurons. And as a result of how much of that channel there is, it changes how excitable or sensitive to its inputs the neuron is, and therefore how much it can listen to, like the hunger neurons and other inputs it might have.
Exactly, exactly. That's it. That's the one layer of regulation. Exactly. That's how these hormones affect the channel expression. So via the basically the gene expression level, and then how hunger affects this ion channel. That's via this neuropeptide NPY that these neurons release, and this neuropeptide binds to certain types of neuropeptide Y receptors, and then there's an intracellular cascade that we don't fully understand yet. That's downstream of this receptor binding event that communicates directly with these ion channels, and it also downregulates the function of this channel. So basically, in a given estrous cycle stage, the hormone ratio dictates how much of that ion channel is made, and it's there in the first place. And then the hunger level dictates how much you're inhibiting those channels that are now present in the membrane.
Right,
and together,
right.
that regulates the excitability of these neurons, so how much they can respond to any incoming input.
Yeah, and so your hunger can change, on a pretty fast time scale. With your next meal, the hormonal fluctuations with the estrous cycle, that's happening over the course of multiple days. And then, of course, so you're talking about hormones, they're changing on a relatively slow time scale. They're getting into the brain, into the hypothalamus, into the nucleus of these neurons, ultimately changing gene expression to change how much of this ion channel these neurons have. That takes time too, right? It takes hours and even usually overnight or over day, if you're a mouse, to sleep and then transport the channels out there and stuff. And so you can even think very intuitively, I think, about how sleep would integrate into this. And maybe if there are parents listening, they can start to think like, oh yeah, when I'm hungry and I'm tired, sometimes I really want to kind of I get aggressive towards my kids. And you can kind of intuit how all of these things tie together.
Exactly. It's very real. I guess we think of ourselves as these very, cognitive, rational beings. But have you ever tried fasting for a couple days? It all goes to pots, and there's not much left of this veneer of civilization. And it's the same with mice, except that we can study how their behavior, can really, flip like a switch based on a very minor and mild, state change.
Yeah, and one of the things implicit in what you said is, so you're using female mice who had no prior experience as mothers, but then you're putting them in front of pups and you're using that as your setup to probe all of this stuff. One thing that's, I think, implicit in what you're saying is the fact that these are naive parents, they're females that have never been pregnant, never had their own pups. That tells us that all of the parental care circuitry is already inside of them latent. It's just sort of waiting for the right stuff to be around, hormones and other things, to express itself, the presence of the pups, the right hormones and the right ratios, all of that. But it's there, it's latent inside of all females. And with that in mind, what can you start to tell us about how the parental care, how the circuitry might change when a female actually becomes pregnant and has her first litter? I would imagine that these hormonal changes are resculpting the circuitry to make it more likely, even when she's hungry, that she's going to engage in the parental care behavior.
Mm-hmm. Exactly. So that's a question that we addressed when I started my lab about 6 years ago. So I when I did my postdoc in Catherine Dulac's lab at Harvard, my sort of project was to really to disentangle parenting circuits in the mouse brain, and I did this. I tried to deconstruct these circuits in both male and female mice. and also, compare the circuit architecture between virgin female mice, so sexually inexperienced female mice, but also moms. And by and large, I didn't find any dramatic differences. And that was sort of interesting because I was expecting, given that the behaviors of these animals was so very, very different. For instance, these virgin females, yes, they are a bit parental, but when you compare the performance to a mother, for instance, it's like day and night. Mothers spend most of their waking hours looking after pups. They retrieve them, within less than a second. It's a very big difference. So I was expecting sort of these major anatomical changes there as well, but I didn't see any. And that sort of, then, I'm prompted to ask, okay, what's actually happening here? Is it that there's finer wiring changes that we can't detect with the methods that we're using? Or is it that there's these more, subtle, functional changes going on that are promoted by these hormonal changes? And when I started my lab, we basically just, embarked on a very systematic investigation of what's happening during motherhood. So when these behavioral changes start appearing and what the underlying neural mechanisms are. And what we found quite quickly was that, I guess, contrary to what everyone was expecting, it's not just after giving birth that the behavior seems to change. So I guess the prevailing model had been that it's the hormonal changes of parturition, of giving birth, that somehow rewire the maternal brain. So we're talking about oxytocin, prolactin, and these kind of hormones. But what we found is when we followed, pup interactions already during early and then late pregnancy, we found that in late pregnancy, the performance, the parental performance of these animals, of these pregnant animals, was already very similar to that of mothers. Yeah. So in very late pregnancy, so pregnancy takes about 20 days in mice.
I see. So before they've given birth, before they've seen the pups, they're already able to behave as if the pups are already there.
Exactly. These are animals that did not previously experience any pups. They weren't exposed to pups previously, so they were sort of pup naive. But by virtue of being pregnant, they already behaved as if they were mothers, and that's that's in a way a quite elegant solution because that's an anticipatory regulation. Because if you imagine, you're a female and you give birth, and all of a sudden there's a litter of 8 pups around. There's not much room to learn. There's not much time to learn how to look after them. They need instant care. And by basically, by preparing the brain for what's happening already during pregnancy, where you have 20 days to basically just, to remodel. That's a very powerful solution to that problem, And then we next ask where in the brain this is happening and what is happening.
Yeah. And so what did you see there?
So what we saw is that that it all a lot of what's happening seems to be happening in the same region again, in the so-called medial preoptic area, or MPOA, which is a key node, as I found during my postdoc in parenting circuits. So we did, in the lab, we did loads of very comprehensive analyses. So we mapped, for instance, throughout the entire mouse brain where hormone receptors are expressed, so the receptors for estrogen and progesterone again, because they are changing quite dramatically during pregnancy. And we mapped this very systematically throughout the brain. And after all of this effort, we found that the expression was the highest in the MPOA, so a region that we had on our radar already. And we also looked at brain volume changes using MRI, so that's just a structural MRI, and found that there are brain volume changes happening as well, and they seem to be most pronounced in the MPOA as well. We don't know what that means, but it just, I guess, it seems to point to a degree of structural remodeling that's happening there. So so that both of these lines of investigation pointed to the MPOA again.
Yeah. Another area that I can imagine here is, so we talked about pup-directed aggression as a function of hunger state and the estrous cycle in parentally naive females. That was the study we were just talking about. And then you just told us about what starts to happen to a mother after she actually gets pregnant and the pregnancy progresses, and then she becomes an actual mother with her litter and all of that stuff. But then after she gives birth, we've got to do some kind of balancing act and integration of hunger and parental care behavior in the natural ecologically relevant state, meaning that right at like the pups need immediate care and attention. They need to drink the mother's milk and so forth. She needs to keep them warm. But at some point, she's got to go out and get some more food and things like that. How much do we know about how that balancing act is regulated in a mother?
still don't know very much about that. So there's a recent study from Mike Crush's lab come out that actually looked at the interactions of these, feeding and parenting circuits as well, and they're suggesting that there's this, this inhibitory crosstalk as well. So if you're hungry, you tend to downregulate parental care somewhat. But still, what was striking in that study is that even though mice are can be very hungry, maternal, they will still very robustly look after the young as well. So they have to be very stressed and very hungry to stop looking after their pups. But mice even 24 hours after giving birth, they already enter what's called a postpartum estrus, and they go out and look for new mates again, and that's why
Hmm.
they're so successful. They can basically they can become pregnant again while still nursing the previous litter.
Oh,
So in a
wow.
way, they're constantly balancing these different types of behavior very successfully. Yeah.
Interesting. What happens? So, how much sort of feedback from the presence of the pups, the sight of them, the smell of them, and so forth, is regulating the new mother's behavior? So, for example, does being around the pups all the time affect how much she's going to go out into the world and look for food and look for more mates? If she's not around them so much, is she more prone to do those things and so forth?
It's a very good question. I don't think we know enough about that, So we know that I guess pups, they're not just these like, passive things that are just lying around in the nest.
Right.
They're actually soliciting the mom's services constantly via these ultrasonic vocalizations that we can't hear, very high pitch, sort of vocalizations that we can regulate the maternal behavior as well. They have certain features, they have tactile features, they have certain, they release certain chemicals that are important for parental care. But that's as much as we know, really. No, we don't beyond that, we don't know how much the pups actually play a role in these behavioral decisions. Yeah, that's
Mhm.
yeah.
What are some of the things that you're working on in the lab today that build on this work or related work on how the brain is managing to read out its internal states? And really, I mean, at one level, what we're talking about is how does the brain decide what to do by integrating all of these different motivational systems together?
Yeah, so I guess one of the obvious next directions would be to scale it up, So now we sort of understand how two of these internal states interact, But what if you scale it up to like 5 or 10 states, Does the same logic still apply? That you basically have this single molecular integrator, these ion channels, for instance, that are capable of integrating, these additional types of states, Like what if you add stress to the mix or sleep? Does it still hold true? Or do you then need to perform a more sophisticated integration where basically, integration of two states is happening in these types of neurons, and the other types of neurons integrate these other types of states, but then there's crosstalk between these neuronal populations. So understanding these kind of principles is I think, on the cards for us. And beyond that, we also want to understand as I mentioned, how hormonal states affect entire circuits. So we, for instance, found out that during pregnancy, these ovarian hormones, progesterone, estradiol, they can rewire this key circuit node in parenting circuits, which seems to be necessary for increasing this parental performance already during pregnancy. But we still don't understand how this works at the entire circuit level. Is it basically that just you have that these hormones act on these very privileged circuit nodes,
Mhm.
or is it that they act on every node in these brain-wide circuits, or are there other hotspots as well? So these sort of the principles of hormone mediated effects on the brain are very much unknown. And I think, in the next few years, we'll be able, conceptually, but also methodologically to really address those things. Yeah.
Yeah, yeah. What do you think? How do you start to think about the consequences of? So humans are different from other animals in many ways, but one of the ways we're notably different is we actually disrupt some of these natural hormonal cycles on purpose for long periods of time, sometimes starting from very early in our development. How does this work start to make you think about sort of the pros and cons of disrupting natural hormone cycles in our own bodies?
Yeah, that's a very, very interesting point. And I gotta be careful here, obviously, being a man, like some of these discussions are probably a bit more abstract to me than they would be for women. But I guess our work has, really maybe, sensitized me a bit to how powerful these hormones are,
Mm.
and how how sort of, thoughtless we seem to be, regulating their levels. Obviously, the pill, has been, transformative, I guess since the 60s,
Yeah.
70s. But
Literally transformative.
Exactly,
Yeah.
exactly. And it has liberated, lots of women, but it's still it's kind of uncomfortable that in 2025 a lot of contraception relies on flooding the body with these hormones that reach, as I mentioned, every a single part of the brain that is in principle receptive to them, and we don't really know, or we're only starting to understand what the consequences of that are. So I think we should probably put more resources into making contraception, molecules that maybe don't cross the blood-brain barrier, or are a bit more, selective in the periphery. So I reckon, 50 years from now, it'll seem barbaric that we ever used these sort of methods.
Yeah.
That's
I think basically what you're saying is that with hormonal contraception, it's having the intended effect on the reproductive portion of this in the periphery by regulating aspects of the menstrual cycle, but these hormones are going everywhere, including into the brain. As you said, they're fat soluble, and they're going to be affecting neurocircuitry, excitability, neuroplasticity in ways that don't necessarily just have to directly to do with what we want them to do for reproductive purposes.
Absolutely. That there's no way they won't do that. If you look at the at which parts of the brain are hormone sensitive, it's probably most neurons in the brain that are sensitive to one of those hormones. So
Yeah.
there are obviously hotspots, but most circuits in the brain will be reacting to these hormone levels.
Yeah. And if we think more generally too, like obviously your work had to do with the estrous cycle and female parental behavior, but these hormones are active in both males and females. There's lots of men taking testosterone today. There's lots of people on GLP-1 drugs. Like a lot of people are taking hormones on a chronic basis now of all different kinds. And I think in general, what you seem to be saying, and what I would say is, we understand one piece of what these things do in a certain context, but these things are powerful. They affect lots of cell types, and we just don't know all of the things that we're kind of mucking with when we do this stuff.
Absolutely. Yeah, absolutely. So I'm really, I'm also, obviously for selfish reasons, but I'm voting for more neuroscientists to go into this area, into basically, combining, what we call systems neuroscience and endocrinology, to not only look at the at the brain as a gland and affecting the rest of the body through hormones, but also the other way around, to see how body derived hormones affect brain processing. So there's not enough happening in neuroscience in this direction, and I really think it's a gold mine, but it's also really necessary moving forward to understand the brain in health and disease.
Yeah. Are there any final thoughts you want to leave people with on this subject of hormonal regulation of behavioral state and behavior, or anything you want to reiterate from what we talked about today?
I can't think of anything right now. Maybe yeah.
Any do you want to point people to your work, your website, or anything upcoming that we might look forward to in terms of the research in this field?
Yeah, so I would definitely I would stay tuned. So it's a more and more active community that's combining systems neuroscience and neuroendocrinology. There's a lot of interesting stuff coming from our labs, but from a number of other labs as well. What will be coming from us is also more sort of developmental questions. We are addressing how these instinctive behavior circuits, how they are shaping during development, and how they control different behavioral repertoires throughout the lifespan. So it's an area that we're also working on at the moment, and I think we have a few really interesting things coming out over the next few months.
All right. Well, this is fascinating work. I love this area in particular. So thank you again for joining me, and I look forward to following along with your work as it comes out.
A pleasure. Thanks a lot for having me. Thank you.
Visit mindandmatter.com johnnykohl.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.